Okay, thank you, Vivian. Okay, all right, I think we're recording. All right, good morning, everyone. Thank you for joining us for this, I guess, open house workshop. So today, our speaker is Dr. Fayuan Wen. Fay is the assistant professor, so she just received her position as sorry in the Department of Biology. So, so she'll be working in the field of computational biology here at Harvard University. So, so she's been applying various computational techniques, such as next-gen sequencing data analysis and then RNA-seq data analysis for for the studies of primarily of sickle cell disease, but also she's expanding now into the field of the HIV research. So, so her, I think your main thrust of her work is to look at the iron overload and its effect on the sickle cell disease patients, using the cohort of patients from Harvard University, which is part of the larger initial BI-sponsored cohort nationwide, which is called TopMed. So she's been successfully utilizing and learning new techniques based on GWAS data analysis and also machine learning data analysis. So, but she also has been helpful, just teaching us and training us to use the Set Enrichment Analysis for the data which we received from the RNA-seq at GW. So, so this, I think, what she's going to present us, and so hopefully everyone can learn that and expand to their own field of research, and I think it'll be helpful, I hope. All right, thank you. It's my little bit longer, but Kifei, yeah, you're on. You can start.
Thank you, Vivian. Thank you, Sergey, for the introduction. Yeah, I think it's good afternoon now, right? Good afternoon, everyone. Thank you for joining me here, and I'm so honored to be invited to give this talk. So in my presentation, I will introduce as an example. So if you want to follow me, I will give some time to do some hands-on to see how we can use this software to benefit. Maybe you have some research project. Okay, so I will share my screen here, and then we'll go from there. Okay, so can everyone see? So can you see the screen now?
Yes.
Okay. Yeah, I see a hand up. Thank you. Yeah, thank you, Benjamin. Thank you, Vivian. Okay, so I use this during pandemic, doing one of Sergey's project. Use RNA-seq. You know, when we do the RNA-seq, we get all the gene lists. So we wondered we wanted to see a gene set enrichment analysis to see what we can find and put in the publication. So I will give you the plan for today, what I'm trying to introduce to you. Okay, so I just think when you can use GSEA, right, in which state you can use. So then I give a little background. Although this method, they get a very huge paper published in 2005. I may not explain very clear why they get this method, but I give basic information for this method. And I will share the data set, is a small set for you to practice and introduce what I do for this data set. And in between what's the software and what the settings for the software, and show you the result look like. And I will leave some time. Hopefully we can do it. Okay, so that's the plan today. And so I don't know how many of you did RNA-seq. You can give me a hand up. So I know. Okay, thank you, Benjamin. I know you doing it. Okay, so I see maybe some of you don't. Some of you, I know Benjamin knows very well all this workflow. But just in case you don't know it, I just give you, before you know the GSEA might be after here, you know, what I'm doing is after here. So I don't know if you can see my mouse. I will use my pointer. So what I'm doing is downstream, okay, analysis. But before, right, you have experiment design. You have either cells, tissues, right, treated, untreated, and you extract the RNA. You have the DNA library you send for sequencing. Then you map your small, like million reads. This is called read small sequence. The sequence is very short. Okay, then you map your reference genomic or transomics that you have a reference. So this color is not real, it's just for indicator for you to understand. So for example, this is gene 1, gene 2, gene 3, right, on the reference. So this sample, the reads mapped so many reads on here. So the table will show something, okay, this is sample 1, okay, for the gene 1, have 8 reads. For the gene 2 have 3 reads, right? So for the gene this purple have 2 reads. So this is a roughly idea for you to understand. Then if you have 2 samples, 3 samples, you can imagine if this have 3 replicates, this have 3 replicates, right? Then you will have 1, 2, 3, 4, 2, 6, right? This is your table will look like. But you remember the reference genome in a human, there was, for us, we have 60,000. We get the name annotated there. So the table will be huge like this. That's what we have at the end. Then for your paper, maybe you will look up, okay, which gene upregulate compare 1 to 2, you know, or 3. So the number will different, right? You want to compare that. But the GSEA is use this big table, okay, is use this big table from the downstream, try to find all the profile, okay, that's when you use it. But this is for RNA-seq for GSEA, but they said for, on their website, for they said for GWAS, if you have plot all the gene list, their molecular files, you can also do GSE, but I use RNA-seq as an example there, okay. Okay, so this is there on their website. I put the website there, okay, and maybe Vivian, you can post them to there to just if somebody can explore. So this is their website, trying to see what's their method, what's their idea behind their method, okay. So they are not looking at a particular certain gene. They consider so the whole set of genes, the whole table you include there. Okay, and you can have a gene set database is a list of genes you want to look at. Okay, basically they will scan to your ranked that big list and get a plot. I will explain later what the result means, but you will run like this. Okay, but what's the file you will prepare to get that to run, right? Okay, that's maybe you introduce, but you stop me if you have any questions. Okay, yeah, I see if you put the chat here. Okay, yeah, so what file you need, right? So this is the thing. Okay, it looks simple. This is the file they require. So yes, you need one file is normalized. Okay, remember the counts file in certain format. They ask in GC4 format. I will introduce you how you get this format, right? You remember this file is huge. For us, we have 60,000 rows for this gene, and depending how many like treatment and groups. So for us, this one we have 36. Like each sample we have 2 runs. Okay, that means that we have 9 case, 9 control, but each one we have 2. Okay, so we have 36 like these columns. Okay, this is the gene ID there. Okay, so they also, you have to tell the software how your experiment setup is, right? You have to tell them, okay, from this route to this 18 is control, you see here, right? Yeah, the other one is the SCD. For us is that, okay, all your same treatment. So this is the 2 file you will, this at here 1, right? Actually need this 2 to be here. Okay, and the other is the gene set database. The database, they have it on their website, and I think I also put in the there's a web link there, Vivian, you can send to them. But so this is the, if you put there, I will show you later on the website. Okay, so they have so many gene sets. Okay, 32,000 gene sets there, and depending what you are studying, you can choose. Normally this one is smaller and is a basic biology gene set. You see, if you study cancer, and this one would be right. This one would be this one is the biggest. I think I remember it more than 10,000 gene set. So depend on what you are studying. You, and also you can download a look there list there. So this is the website there. Okay, and you can also, if you don't want the public available gene set, if you think I'm interested in a small data set I'm studying, like in our lab, in SCD lab, we have a gene set. We want to see the HIV restriction factors, right? So we have a long list along here. I didn't include all of them, right? We have a long list here. So you can just generate, they call it GMX format. Actually easy. You have a name, okay, header. You put NA, I don't know why, but you have to put there. Then you put all your list. This is easy for the gene set. Then you, when you run, you basically have 1, 2, 3 files ready. Okay, or you don't have this file, you use there, click on the software, choose one. Okay, so that's the file you will need. Okay, so any questions before I move forward? How the result? Okay, I think you're good. So the method there, the huge paper there, and tell you, you know, how they design this. Okay, so I just gave a quick view there. You know, this is the big table, right? From RNA-seq, they label like this, but you imagine because the table is so big, they just so for example, you have a treatment or control or treatment, right? So they rank their gene, but how they rank their gene? They call it, they use a method, a metric, depending different data set, but they call it, they calculate the signal to ratio, signal to noise ratio. I don't get a full understand because this paper I didn't read all through, but they ranked your gene. You can imagine the 60,000 genes they ranked, use their organism ranked. And you have your own gene list, right? What's the name there? So the software do is they will, okay, for the first one, they will go to look here, right? To look, is there one? Okay, go look. Each one to scan them. Then they will calculate, they call it enrichment score. But it just depends. If they have in the middle is a cutoff, okay. Anyone on the top would be positive, on the bottom will negative. Okay, so for this example, you will see, okay, on the top there are a bunch of genes. Each line here is a one gene name. Okay, they just indicate like that. So enriched on here. And depending how you set up, they will say, if you say here is a correlation with A or B, I will show the result. You will see, okay, this bunch will maybe correlate, this just show you correlate to what. Okay, so basically you will get a graph like this, show you your enrichment score goals depending on your gene list. I don't know if you have any questions, you can stop me here. Okay, yeah, so this is the method there. Okay, so for our study, I will just give a quick introduction for our data. Okay, so we use PBMCs of the sickle cell disease patients. Okay, I think the whole idea at first is we won't get HIV, some blood to work with, but is we think, okay, maybe we can mimic them in the PBMCs, this tool, you know, to study them. We use one kit to have PHA and IL-2. So I read them is try to active their immune response. You can understand that way. Okay, so we have 9 control patients and 9 sickle cell patients. Okay, so basically you have you can think this is treated and treated together is a control and the sickle cell patient. So this is the setup. Then we send for sequencing at the CFAR for sequencing. I think Sergey did this part for this part, okay, quality control, and we use the Illumina, the website for the drag in RNA and differential expression, that software online, to get this table, okay, to get the raw count table, like a huge table, right? I just use the small genes and samples, right? We have more than this 6 samples, but this is just illustration to show you. Okay, so this is what our experiment, go. Then I will show you the data and I'll show you what the results look like. Yeah, so we then this is the raw. We have 2 files to generate, right? One is the raw count data from what we have. You have this many rows. You see, I just show you 15 here, right? But you have 60,000 rows, and you have, because we have 36, right, runs. So this 36, then plus this gene ID here, so we have 37. So this is a huge data there, right? So this is the raw counts, right? So you see there's no gene ID. The other is you generate is a differential gene expression file, right? This file is also have this many rows, right? This many rows, and also, but you see this they based on your raw counts. This is when you do RNA-seq, you will want to log 2-fold change. You will have a cutoff, right? You want a highly significant upregulate or downregulate, right? And the other one you want for your GSEA, you want to get this column, right? You get this column, put in your previous this column to match your this ID, right? So that's the only information we need here, okay, to put in previous file. Okay, so that's the 2 file you use, right? So in order to generate the input file for the GSEA, right, you need this raw count data, right? I showed you there. And you also need this file. Okay, you have the count file, right? Normalized font file. So you need this file. This file called metadata file, right? Basically, also you see here, this is the column, the run 1, run 2, run 3, right? This name. So basically, you here run 1, run 2, run 3, this name. You tell them, okay, what's your group is. This is you tell them, is this SCD or not, right? You saying this is your replicates, how many? You need 1 to 18. So this is the format. So you have this. But this part I have to you use DSEC in R and TeleVas to input these 2 files, get the formatted file. They call the normalized. The normalized step you have to do here. So I don't think we can go through that part if you want to do the hands-on, because I didn't get a smaller data set for you to play with, right? And but I can share with you if you want to format your own data set. So this is the instruction on the DSEC website. Basically, you have to load your DSEC2 Tediverse. This is the basic 2 packages you have to use. Then you get ready for your, I think, the raw count data and your metadata, okay, to load in and run the program, let them change the number to what, okay. So I will show you how the numbers changed, but this is the 2 files. The first file, right, is from the DSEC. The second file is you can manually, I think, work on it, right? You just label it, where is your control, where is your treatment, right? So, and I will see you how, how I will put in a, just see in Excel is easier, because this part is all headers, right? It all clumps together. So I will put in Excel, just show you the first one where you should pay attention to. Yeah. So for the file 1, the normalized count file, right? You remember this gene ID is from your expression file, this column, right? So this huge file is still have this many rows, right? And have this many columns depending how many runs you have. So you see, the gene ID here, the common error when you put GSEA is because your gene ID originally there have a version number. They might have a version number, something like that. You have to get rid of this. Otherwise, it wouldn't run and tell you error. So you make sure delete that. How you delete? I just found it very. In Excel, you can put okay dot star and replace with what. So I want to find another way to replace every you change it to the you get rid of this version number. You can have your own way to do it, but you make it clean like this. Okay? So this is the first common error when you input if you don't have this format. The second one, you make sure okay this number is exactly as how many gene ID is. Okay, this is the other explanation. So the third one, you also make sure this number is exactly how many runs, right? How many sample, how many runs you have on the table. For us, we have 36. Okay, so this is the main things. And also each one, I keep the original, the run name. They are unique. If you want to rename it, you make sure you are not saying control, right? You have to maybe say control 1 or 2 or 3. You make them unique. So that's the main, when you do it. But you see here, before was the raw count don't have this complicated, decimal numbers, right? Now it's just because it was go through DSEC, they do the normalization, make it like this. So this is the format for the first one you have to pay attention to. Okay, for the second one, it's a little easier, right? You just tell the program how you set up your group. Okay, so you tell them I have 36 runs, I have 2 groups, but here in the text file would be here is a space. Okay, here also a space. Okay, so make sure that that's it. And then have 1. You just tell them, then have a pound K space control and a treatment, depending how you name your 2 groups. Okay, then you give this is one. If you open Excel, would be one long row. Okay, the SCD would be here. Okay, it's just because I opened in a text file, it should like this. Okay, SCD will be here, so it's a long one row here. Okay, one row here. Okay, so this is the second file there. Okay. So and how you run it, this is the software look like. So you first can load your data, then you run. That's it. But for this software, I just want before this morning I double check how many like space it takes when you install it. Maybe for Mac, it looks like the file takes 160 megabytes. Okay, when you run it, how many memory requires? I checked up. Looks like depending what the data gene set you will use. So when we practice, we choose the first Helmac one. They have a smaller set to use. So for me, when I run the immunology, that data set there, that maybe have a few thousand gene set. So it takes 2 gigabit like memory, something like that. So and also takes so many minutes. Okay, for when you load the data first, okay, you can browse the file, you can load from before where you used. So I will show you in the real. So then you browse your file, choose the file, right? Then you just load it. Okay, so it's straightforward for the software. Then for the settings, you just say I run it, right? After you load it. So you choose your normalized count file, you choose your gene set. They see the drop down menu, there are so many, I will show you. Okay, so you choose whichever you are interested, or you can upload yourself, and you choose the number of permutation. They suggest 1000, but for our practice, I think we use a smaller number just for you to get a quick result. Because if the number higher takes a much memory. Okay, but they suggest 1000 maybe have a more stable estimation for your results. So and then you drop down, you choose your phenotype label file. Okay, So then this cheap platform for us, we use human ensomal gene ID. This one is also a drop-down menu. You click it. So the other one, I will go default, and you can name your folder, like the result will, what the name is, and you can choose where you want to store your results. Okay, so by click this drop down menu. So this is straightforward software. Okay, after run, before it was green, now it turned pink. It's a newer version, it turned pink anyway. But the success, if you click success, it will turn up like a web page, look like, for you to click all the link, get your results. You can also go to your folder where you store it to go through. It's a huge folder usually. Okay, so when you set up, you click this run, when success will be here. If something wrong, it will go error here. Okay, then you click here to see what's wrong. And usually the format was not correct. Okay, so that's the software here. Okay, so I explored things, why I list here, just thinking, if you have different studies, it can build up very quickly so many things that you can think of. This is two, right? This two. If I have this two RNA-seq study, then I run this gene set, and I run this gene set, right? I run this gene set. So there's already six big folders, and you have to, and each one, this gene set, right, each set have hundreds, right? So you sometimes need some your knowledge to your background, in your area, to decide what result you really want, right, to see. So I just demonstrate this one just because of the immune response, right? So I use this one. That's the result. When they come up, we will give you a bigger, this we will a summary. But in the summary, they have the links for the table, the graph underneath. Okay, so enrichment in a phenotype, right? In a control is how many gene set, and they have a snapshot of all the pictures, and they have TSV files. They also have a web page files. Okay, and in a phenotype, how many enriched, and what's your data set. So this is the summary, and go down. They also have a heat map for your first 50 ones in your data set, like a summary, but just in their ranking, right? First 50. So this will be showed, okay, in your control, it's upregulated or enriched here, but in the treatment was downregulated. So this group of gene was downregulated in the treatment. So things like that, the result. So if I click here, right, enriched result in TSV, like here. So I will see a big table, a big table. 3,000 some rule is depending how many gene set. Okay, because in that gene set they have so many gene set. So each rule is a gene set. For example, first one is lupus CD4 T cell versus lupus myeloid, right? This one. So if you want to go see, okay, in this gene set, which genes are enriched, you click the details there. Okay, it will give you the specifically for this gene set what's going. Okay, they also give you the false discovery. This is what you want to look at. Less than 0 to 0.25 considered significant in their method. Okay, so so if you click the detail, it show you this result in this genes, in the first gene set. Okay, this is also a long list. I screenshot part of it. Okay, so this list corresponded to this graph. So like saying, okay, this TST, it should be this green bar. Okay, this one, it should be this bar. Okay, so it just means this bunch of groups, right, was in a dark area would be this gene. Okay, So this will be just here you know the all enriched score is all manners, right? So it's here, right? So this group of this is positive correlated is this field, okay, and here it negative correlated with SCD. So this is the how the graph will look, and also this color indicates what their correlation is. Okay. So this is the graph. If we look, yeah, they also down that details, they also get to you in this gene set, in your data set, what's your genes is upregulated or downregulated in your control and your treatment. So this is how they look, and they also produce enrichment score distribution. Tell you in this data set there was all in the manners range, right? So if it's in the control, you clicked, right? You clicked on the main menu, the control one, you give you the list. This is so many gene set enriched. And if you, the same thing, if you want to see in the detail, you see the information there. So if you are specifically introduce one, you are encouraged to have your own. Otherwise, there's so much information for you to go through each of them. Okay, so if you, okay, for the first one, for example, right? So this is the data set looks like. So they saying, okay, this bunch of gene at here was enriched in the control, okay, and this bunch was positively correlated with control, and this a few on the list. This is the graph. This is the table you really want to look at. Okay, so this is the result how look like, and also the same thing they produced for this data set was a heat map in the data set look like this. Okay, so I will just stop here for my quick introduction. Maybe we can go through on the website if you want install them and have a quick idea, how it look like. Okay, so I here the one of two files, small, not small, but it's just subset of a data set for you to practice if you want to follow on. Okay, so I will stop here. I really appreciate CFAR invite me to have this presentation, and for the past 5 years I've been working with Sergey in Sergey's lab do the whole genomic sequencing and part of NGS analysis. So even though now I joined the biology department, but I'm still working on some of Sergey's project, and appreciate the lab member support there. Okay, so now I think we move on to the fun part. So everyone getting a little, hopefully, I don't know if we get some errors, we have to troubleshooting for a few days. Okay, but I will end you now. But before I end, is there any questions so I can go back to the slides?
Yeah, anyway, thank you, Faye. Yeah, I just would like to comment, right? It actually is much easier than it sounds. I think the critical part is, as Faye pointed out, is to make this normalized data sets after you have RNA-seq or otherwise, and then you just play with this program. So I think the advantage if you do it yourself is that you can actually tweak it the way you want, because otherwise, if you ask someone to do this GSEA analysis, they'll give you some sets, you don't know what to do with that, and yes, so it's a bit like time. But I think what probably people will need, I think, moving forward, like this, if you don't do R, so I mean we can share the script, of course, but so maybe help with the normalization could be helpful, I think. But once you have normalized set, you can actually play yourself. It's very easy to make your gene set as well, as Faye pointed out. Just a file, and then you can upload it. And then there is also one thing, right, Faye? If you have not so many samples, then I think there is there are some tricks how I guess, set up the parameters so that it won't kind of blank the analysis. So anyway, but that's only. So if you have many samples, it's really not a problem. But if you have like 3 or less, then I think the statistics parameters should be reset. I think. Yeah.
Yeah. Thank you, Sergey, for the comment. Yeah, and Sergey first let me try this. Then he picked up. Then he picked up the R to the normalization, and he did his own analysis for a few later on project. So just because when we hear the result will a few hundred megabits. You know, I usually put it in a Google Drive, let show him, and he still have to go through them, and would rather to run it quickly. You know, I will just end the show here. No more questions, right? So I will end the show here. Okay. So so Vivian, did you send the Google Drive, right? The first one, the Google Drive.
Yes, it's now in the chat.
So yeah, so I will show you the files, and then we go to the website, and then we start from there. Okay, so I will stop.
I can see
there. So this, when you when you click the Google Drive, the file you can put in a folder. This is the two file I shared with you. So firstly, the normalized count, right? The other is the sample. This one ignore it. Just this morning I ran it. The result come here. Okay. So this is the normalized count. This is sample phenotype. If you not sure, you don't try. If you really want to open it, you can open with, right? Usually I don't open. You can open in a terminal, maybe easier. But if you really want to open, you can. But I not encourage you open with Excel. You can see it, but it's the what I showed in the slide. Okay, so these two files you will have an input file for your GSEA. So let's see how you go to the where is the downloads. Okay, so this is the website. I will show you, right? You click one of the links. Is the hopefully you're already there, right? So this is their website there, and this is their home room. Just give you introduction of their method, right? So I use this picture, and you can go here for downloads. And this is their GenSet database introduction. This is the documentation tell you how you go. So I go each section, then we can see the downloads, right? So for this molecular database, right? Okay. You see, this is the database I mentioned there. So each of them, and they also have a mouse for the mouse one, right? If you work with human, this is human one. So this is the oncology gene set. So this is the smaller one. So if you click this, right, click this here, it will go into another one thing. You can download them, right, to see what's in their list there. And I think usually in the software you just click, you don't need to do here. But if you the GSEA, the other version is you work directly in R. Okay, so I think people will download that to work. But for us, if you download the software on your computer, you don't need to download the gene set. But just if you want to know more information, this is the data set. Okay, the documentation part. Okay, so for how you run it, right? User guide is here. Right, user guide is here. Just tell you step by step. You have to figure out. And DSEQ and RNA-seq, right? This is what we are using, right? Using RNA-seq dataset with GSEA. So this is the documentation. They tell you how you do the. It looks easy, but for me, I was to go through on their website, DSEQ's website, to go step by step to see how you will do that part. And so that's their documentation. If we go back, right? So if you click, I think that's it. I think we have another website I want to show you. This is my results there. I want to show you the another one anyway. So the manual is more in detail, but anyway. So this is the downloads, right? If you click the downloads, it just show you, okay, you can download for your Mac, you can download for your Windows. I think most of us use Mac or Windows, right? Depending what, but let's just show you if you want to really follow along. If you say I want download, so you for the Mac one, you download here, right? For me, it's going to jump up here, right? So I click it, so it will unzip it. Okay, it will unzip it. So and now goes to my downloads, right? Where's my downloads? Here. So you see, because I click it, it's unzipped already. So the easy way is you grab to your application. Because I already have one, I wouldn't grab there, but if you see, ask me to grab. So you grab to your application, then it will be there. You can directly use it. So that's the the thing is already, you know, being there. So then you go to your application or launch bar for, you know, to open up. And for me, I will be here. So I already open up. I don't know if you, some of you try to follow on, if you get it installed or not. But so this is the, you know, after you install it, will be here. This morning I tried to run first one. I forget to attach this data set, get error, but second time I make sure it was there, so it will run. So is there someone need me to wait a little second if you are installing, or you are good? If I didn't hear anything, I will just okay, so I think, okay, so nobody have any comment thinking I should wait a little second. So I see there are 2 new messages. Okay, here the link. Okay, so then for the first part, right, I don't know if you are following or you are just looking. You looking is okay. Load the data. Okay, so first part, you load your data. You can browse for your files. You can load where you last used, right? So for browser file, for example, right, and because I already, you can go, for me, this morning I set a folder for our own purpose, right? So this is the file. So I think I want import these 2 files, right? So you click open, it's saying load 2 files. There were no errors, but sometimes they saying error, no loading. So you will have to go back to see what's wrong, and there's no hint for you to tell what's wrong, right? So the only thing is that you have to go back to their documentation to make sure your format was correct. I think that's the main issue. So, but there's okay, you say okay, it's all done. So I think the file is here. After you put your window is here, okay. So the normalized count is here, right? You can grab here and some sample phenotype, and you can grab here. So that's the 2 files, okay. So you see, I want to load these 2 files, right? So it already loaded, but why tell me again it's loaded there? Application. Okay. So let's see if we can run the thing is run loaded down, right? So I will say here run the GSEA, right? Oh, it's already, I should clear them up, but let's see. You can choose the normalized count is where you get a big table, right there. And the gene set is, if you go here, there's a big drop down. It's on their website. That's there. If you don't know which one you use, right, you go to their website to see which one you want to use. And you can also upload one, which we didn't do, but you can upload one. Then after you upload, your list will show here. You can choose them. Okay. We choose the first one just because it's a smaller data set, easier to for the computer to run, okay, to finish it. Okay, and it's a basic called hallmark, this one. Okay, then you say yes. So also this number you can use 1,000 for your own task, but here I use 100 just for demonstration for quicker. Okay, and then you label, right, you choose your source file, this file, right, and you have to say I want to compare control with versus case, or case versus control, right? So either way you want to have your graph to lay out. So this is right, and so I think I keep this default there. Usually we use the gene set period tab, and the gene keep tab. You find the human insolvent ID, this one. Okay, so we just use this because we have the insolvent ID on the first row, right? Then you give it the name. You give a name thing. You give them name thing. Maybe okay, so I just want this, right? So for my, so just I usually give a date is because something happened. I read my notes. I know what I'm doing to, something I can dig back there. Okay, I keep the other all default, but this is where you want, right? You want put your, you can think I want to put it in this folder, right? I want to put it in this folder. Okay, so you put it in this folder, then the other one keep default. Okay, keep default there. Then you can run it. Say running. But sometimes have error. Then you have to stop to go back to see what's wrong, right? You can. So I think this task you see here when it run, it's saying give you saying done or reports. But this is quicker, right? This is just because I choose a smaller gene set. If I choose the immunology, that one takes half an hour. Okay? And so this is the memory they are using. So when it's done, right, you click the success. Or you can go back to your folder. There's a folder created. Okay, so you click the success. This is what looks the result. It's in your folder. You see it's in my folder, documents in this folder, right? So this is the and the file in your folder. If you want to look at later, right? So the file in your folder, it will be index this file. You click up, can link to everything. Okay, you can click the first one, give you all the gene set, the graph there. And if you click the result in this HTML file, is also giving, okay, the first gene set. Let's see the first one, right? The details will give you all the graphs and all your gene list there. And you see, I just keep 3 cases, 3 controls just for practice, right? And so this is the gene map there. Okay, so this is the enrichment score distribution. So this is the result, right? Enrichment in phenotype, you can do the same thing. Click. Oh, this is will. I think it will looks like it just download for me, but okay. So this is each one them. You can go through them to see which gene you are interested to have, right? You see here have a warning. It's just because some. They said that we have duplicates in the data set. Okay, so I think I will stop here, right? So the big heat map for what they are thinking in their ranking will be like this. Okay, then I think this at this stage, you will decide, okay, is there something you are interested in this data set, right? Okay, so that's the big, and if you want to see in your folder, it will be look like this. So see, it's created another folder. So the folder is so huge. I mean, so when you look like it, because they put everything. So I recommend the same thing is you go down. This is the index file. Okay, the same thing. We'll link everything so for you to easier to look what you want. Okay. So I usually after I get this, I will put the big folder in a Google Drive to share, to share with the colleagues and Sergey. So this is the okay. So I will stop here and So if you have any comment or questions, I'm stop. Thank you, everyone. Thank you for your attention here.
Yeah, thank you, Faye. So again, I think my suggestion would be this is going to be online, right? On YouTube. So I think it's going to be posted. So the easiest way is you just put this recording on if you would like to practice, and then you just kind of keep going step by step. Also, there are some as YouTube videos. If you find something is not very clear, I think it's also possible to find them. If you need any help, further help, you can just let us know.
Yeah, I think that
She's the main she's the main driver.
Yeah, I think today, because the time limit, I didn't show if you want to work in R or you want to, install R and R Studio and do the DSEC. Actually, it not take that time when we have the script, right? You just click the button, everything done in minutes. But I just thinking if people you don't have the R Studio, and I don't, if you are interested, I can set up a small data set for you to practice. Okay.
Where will the recording be available?
We'll be uploading the recording after this session on the YouTube, and we can also distribute that out to everyone who's participating
The
today.
link, right? Yeah, and the okay, that sounds good.
Thank you, Namita.
Yeah, because until we practice it, it's very difficult to do anything.
Yeah. So after you get the correct format, when you run it, it will pretty easy. But there will be some errors, at the first few run, because the formatting. Yeah.
Yeah, we'll see if we can ask you to set up a small practice thing once I go through this, right?
Yeah. Sounds good.
Thank you.
Thank you. Any more comments or questions? Maybe I go a little quicker, or I talk too fast.
And also, I think these days you can. I mean, if you need help, like with the script, for example, it's very easy to go to ChatGPT. To tell you.
Yeah.
Most of the time, okay. I think sometimes it gives you like wrong script, but you just shoot it back and say, okay, the script didn't work, and then so
ChatGPT is a very good tutor. Yeah, as long as you have the R Studio set up, you say how can I do formatting my ESEC normalized count data. I think it will get you through.
by contactor, yes.
Yeah.
I think we are at the stage where I think many of us will just have to use it, I guess, routinely. Because it's ChatGPT is no longer free, but all the scripts are. Yeah, we can share. I think, and the R scripts are free, so you can just download whatever you want.
ChatGPT is not free?
I think you have to pay some. I pay just a bit money for that, but maybe it will be more expensive as it goes. But I think you usually you given like a couple months, I think, of like free ride, and after that it will start charging you. Depending what you ask it to do. I mean, if it's like small thing, it's very little money. But you have to be careful now when you're going through the NIH study section orientation. They tell you never use ChatGPT for any comments. Basically, whatever you put in there, it's not secure. So if you put your like proprietary data in there, that's it. So you cannot be sure that it's not as secure. So, but if you ask for script, yeah, it's fine, totally fine. Yeah. Right. I don't know if you have any more questions. Vivian, maybe we can do the after that like follow-up questionnaire. Just feedback, and then see if people like it, or what needs to be done to kind of to improve this type of workshops.
Okay, that sounds good.
Okay. I guess if there's no more questions, right, we can probably stop. And
I have a question.
oh yeah, please, yeah.
So it's regarding the normalization of the data. So when you went over, how would you normalize the data?
Yeah, that's what we are talking about it. We use the DSEC tool. That's the software. That's the website that they recommend use that to get that part. So I just have one slide right here. You can go to the DSEC tool to follow the instruction. If you have some troubles, maybe you can ask for help. We can send you the script and maybe can get you through. Yeah, it's the R package. So I was thinking today to get that to do the hands-on, but I just think the time not. If I should maybe next time we can planning to saying before this seminar, people can install software to know what's going on, have a short document. Then then when in a session they can do the things. So.
Yeah, I think it's geometrical mean normalization, whatever it means, right? So if you do your, unfortunately, if you do your run in Illumina, they'll do that, but they won't give you like they only give you like raw gene counts, and it won't give you the normalized gene count. So you'll have to normalize yourself. They'll give you like I think they'll normalize and then give you like summary, but not like individual gene counts to be normalized. So those counts needs to be normalized. But as Faye said, we have a script, so we can share a script and we can run it. Should be straightforward.
Thank you, Marima. That's great, good question. That's the key part actually. So.
Yeah, absolutely.
Okay. Yeah, Ulahan saying, thank you for the explanation practice. Did you do the practice, Ulahan?
I think my final comment. There are lots of links right in the chat, so I'm not sure what we should do with that. Maybe
I can send those as well.
Yeah, maybe you can put them together. Yeah.
I couldn't because I'm in in the hospital's computer. I couldn't download, and I can't do that. So if you knew it beforehand, then we have to go to IT and get it approved and work on the computer. So, but I can try it in my at home on my Mac. So,
okay.
Thank you.
Because I have the file, right, the red format. So in my end it works. It should work if you have the software. Yeah.
Thank you.
And Fayuan, would it be okay if we shared the data set with everyone via email?
For me, it's okay. Sergey I rename the sample name. I just have 3 columns for each, 3 run for each. So it's okay for you.
Yeah, I think it's truncated the data set for the practice, so I think it should be okay. I think as long as it's truncated and just keep in mind that it actually is our working data set. So just don't use it for. I mean, if you want to use it, just talk to me. I'll be happy to cooperate. But just don't use without our permission. Like for the research. For practice, no problem at all. Yeah.
Okay. Great. Well, great. Thank you all for joining. I will follow up via email with the resources, the sample set, and the evaluation, as well as the recording for future reference.
Okay.
Thank you again. Thank you, Fayuan.
Thank you, Vivian. Thank you, everyone.
Bye.